Hello children, welcome to biology class. Let's discuss the topic sex determination from the lesson Principles of Inheritance and Variation. Children, in the year 1891, biologist Henking observed a trace of specific nuclear structure in few insects. He also observed that the specific nuclear structure is located on 50% of sperms only. That means the remaining 50% of sperms are devoid of this specialized structure. He called this as X body, and he was not able to explain its significance. But later, you know what happened? It was observed that the ovum that received the sperms with X body become female and those not becomes males. So this X body was called as sex chromosome and other chromosomes are called autosomes. This is a chart of humans' chromosomes. 23 pairs of chromosomes are exhibited here, and from this diagram it is very clear that 22 chromosomes are autosomes. See children, these 22 pairs of, that means in total 44 chromosomes are autosomes and remaining one pair is sex chromosome. Okay, if it is X and Y, it become male, and if it is X and X, yes, it become female. Now let's analyze what are the differences between autosomes and sex chromosome or allosomes. That means chromosomes that do not determine the sex of individuals are autosomes. Chromosome number is 2n-1. Their morphology is similar in male and female. The number differs from species to species. They do not exhibit sex linkage. Whereas in allosomes, chromosomes that determine the sex of individuals, chromosome number 2, morphology is different in male and female. Each diploid organism usually has 2 allosomes. They exhibit sex linkage. Now, we are going to analyze the sex determination in insect. Children, we know that in humans and other organisms, XY types of sex determination is seen, isn't it? But in some insects like Drosophila, X0 type sex determination is present. And this XX represent female, same as that of a human being, and this X0 represent male. And which all gametes will produce from XX? Only one type, isn't it? Whereas the male members produced the gametes with X chromosome and 0 or O chromosome, and in fusion those organisms produce XX will become, yes, female, and XO become, yes, male. Children, in both types of sex determination, male produce two different types of gametes, isn't it? In the case of human being, which are the two different types of gametes? Yes, X and Y. Here in Drosophila, X and 0. That's why this type of sex determination are called male heterogamety. Now, let's discuss the condition, that means the sex determination in birds. Here, ZW type of sex determination is present. One more specialty is there. Here we can see female heterogamety. You know what is female heterogamety? Instead of male, here females produce two different types of gametes. Clear? So ZW, female gamete will produce a gamete with Z chromosome and gamete with W chromosome. Okay, but male produce only a single type of gamete, that is Z chromosome, just opposite to that of human condition. Okay, so this is female heterogamety, whereas in human being and Drosophila they exhibit male heterogamety. Okay, now let's discuss sex determination in human being in detail. I know you are aware it. We know that out of 23 pairs of chromosome, 22 pairs are exactly same in male and female called autosomes. A pair of X chromosome is present in female and in male X and Y. So during spermatogenesis, male produce two type of gametes, sperms. 50% carries Y chromosome and remaining 50%, yes, X chromosome. Whereas female, they produce only one kind of gamete or ovum having X chromosomes only, isn't it? So when sperm having Y chromosome, the sex of the baby is, when the sperm is having Y chromosome which fuse with the or which fertilize the egg, the baby will be male, and when sperm carrying X chromosome fertilize the egg, the sex of the baby is, yes, female. Now you just observe this diagram. Father, X, Y, and they produce two different types of gametes, X and Y, whereas mother can produce only single type of gamete. And during fertilization, fusion, what happened? XX become female, XX become female, XY become male, then XY become male. That means there are 50% probability of a female child and 50% of probability for a male child. So I think the topic is clear to you. We discussed sex determination, human being, XY, XX type, sex determination in Drosophila, XX serotype. Both Drosophila and human being exhibit, yes, male heterogamety, and in bird, ZWZZ, they exhibit female heterogamety. Clear? Now let's discuss next topic, that is mutation. Mutation in single term we can write it is a sudden inheritable change. Okay, sudden change that change will be inheritable to next generation. Okay, so there are three different types of causes. Physical cause such as X-rays, gamma rays, infrared rays may alter the manipulation of gene. Then chemical causes are there. Some chemicals may alter the genetic manipulation. Then some viruses and bacteria may alter the genetic manipulation. So mutation is a phenomenon which result in alteration of DNA sequence and consequently result in the change in the genotype and phenotype of an organism. Okay, see children, it results the change in genotype and phenotype, visible change, that is phenotype. Genotype is change in genetic manipulation. So mutation is possible to produce change in genotype and phenotype of an organism. So the mutation that arise due to change in a single base pair of DNA are called point mutation. I will exhibit a diagram to you. See here, glutamic acid and valine, two amino acid you can see. Glutamic acid is produced from the genetic codon GAG, whereas valine is produced from GUG. A single base pair change is there. Instead of adenine, when uracil came, instead of producing glutamic acid, the amino acid valine will produce. See children, here in the 6th position, instead of glutamic acid, if valine produced because of this single base pair change, the entire protein will change. Here, this amino acid chain will produce normal red blood cells, but a single base pair change results the production of sickle-shaped red blood cell, and this condition is termed as sickle cell anemia. See why the sickle cell anemia is produced in an organism? A single amino acid change, a single base pair change. This is point mutation. So different types of mutations are there. So in our textbook we have to discuss about only a point mutation. And example is sickle cell anemia. Clear? Next topic, pedigree analysis. Children, do you know what pedigree is? I will show you some of the pedigree charts. This is one pedigree chart and this is another pedigree chart. Children, this is a pedigree chart of Queen Victoria. Now, can you explain what pedigree is? Yes, to explain or to analyze a single trait or a disease condition or an abnormality in different generations of a family. We can use pedigree analysis. See children, the analysis of traits in several generations of a family is called pedigree analysis. The inheritance of a particular trait is represented in family tree over several generations. It is used to trace the inheritance of particular trait, abnormality and disease. Here in this pedigree of Queen Victoria, the disease hemophilia is exhibited. Okay, so in coming classes we will discuss more about different types of disease conditions and their pedigree chart. Before that, you should study the symbols which is used to draw pedigree chart. The square represent male, the circle represent female. If it is shaded, it will be affected individual, that means disease causing individual, and between square and circle, if a line is there, mating. Then again we can represent next generation by using this line. Then identical twins, non-identical twins. The diagram is there in our textbook. Okay, you study children. Okay, so that's about today's class. Read the textbook thoroughly and study well.